one of the main reasons is that NR and NMN are simply not addressing the root causes of why NAD is actually declining in the first place. So usually when something's declining in the body, there's a reason for it. And what NR and NMN do is they don't look at what that reason is. They just go, okay, just put more raw material in and hopefully we'll get more NAD out the other end. But what we now know is a lot of studies have demonstrated that there are two main things that are going on in our body to cause NAD decline. The first is that our NAD production and recycling pathway in our cells declines with age. So what happens is that when NAD is used up during repair and things like that, it's actually broken down into precursors, and the precursor it's broken down into is nicotinamide. Then what would usually happen in our healthy young cells is this nicotinamide would literally be scooped back up in a pathway called the salvage pathway, which recycles this precursor straight back into fresh NAD again. So it's a kind of clever pathway because it means that as NAD use goes up, there'll be more breakdown products, and this pathway gets turned up and it recycles it back into more NAD to fulfill the demand for NAD. But what we now know is that as we become older, this pathway declines. So specifically, there's an enzyme in this pathway called NAMPT. This is the enzyme that declines, and it's a rate-limiting enzyme, which basically means the level of this enzyme directly dictates how much NAD will be made. So if the levels of this enzyme go down, you'll get less NAD. And we find that levels of this enzyme do decline during aging, which means that NAD simply can't be recycled as it's being used up. And this is why you begin to get a deficit. So if you look at a precursor like NR or NMN, it's ignoring this problem. So it's kind of like if you think of a factory, if the cell is a factory, you are saying, oh my goodness, production's gone down in the factory. How do we fix it? We can see all the machines are broken, but let's ignore that and let's just ship more raw material in and hope more NAD comes out the other end. That is essentially what is happening. So the other thing is we now also know that that's not the only problem. The second problem is inflammation. So chronic low-grade inflammation, we all suffer from it as we get older, and we know that it causes increased expression of a protein or an enzyme called CD38 on our cells. And CD38 uses up NAD. In fact, it's a prolific NAD consumer. It uses so much and wastes it to drive further inflammation. So again, we know that we have more CD38, which is using more NAD. And again, if you think of precursors like NR or NMN, if you are boosting your NAD levels, but you're not inhibiting CD38, then it's likely that a lot of this NAD that you are increasing is actually getting used by CD38 rather than other important processes like DNA repair enzymes, which also use NAD, or the sirtuins, which are crucial longevity genes, which also use NAD. And the reason we know this is because it's been calculated that CD38 has a higher affinity for NAD than the repair processes, which basically means that if NAD is available in the cell, CD38 will grab onto it strongly and use it before any of the more beneficial processes even get a look in. So, you could be thinking, oh, I'm doing this good thing, I'm using NMN or NR to boost my NAD, but if you've got high expression of CD38, that NAD is actually driving inflammation. So by not fixing the root causes, you can be actually making other things worse. When things like NR and NMN were really just emerging, and even at that time, I was looking at NMN and NR and saying, well, it's great that there's this data, but it's also becoming very clear that they're not addressing the root causes. So, if we're going to develop a product, we want to do, the next generation of NAD supplements. We want to, bring something out that reflects the latest science. So what we did was we designed a product that not only had a precursor in it, but also had lots of different other ingredients in that were all designed to fix the root causes of NAD decline. So interestingly, a lot of people say to us, well, why have you not used NR or NMN as your precursor? Because we actually use something called nicotinamide. And the reason we use nicotinamide is because nicotinamide is actually the best precursor in terms of bioavailability, and it's the precursor that our bodies naturally use to make the largest amount of the largest proportion of NAD. So that's the reason why we use nicotinamide. It doesn't require any transporters to get in the cells. It freely diffuses across membranes, and we know it where it gets to where you want it to get to in the body. Unlike NR and NMN that do require transporters, and there's also a huge amount of debate about whether things like NR actually just get metabolized to nicotinamide and then taken up by the cells anyway. So we just sort of stayed out of that whole argument and just went straight for the nicotinamide because it does work just as well. We then were like, okay, what are the main things that we want to address here? So we want to switch back on NAMPT. So we have several ingredients in there that do that. We have an ingredient called rutin, which contains an active called quercetin, which is an NAMPT activator. We also have alpha lipoic acid in there, which is also acting to induce mimic the effect that you have when you're fasting, to again switch on that pathway. So we see increased levels of NAD salvage and recycling in the cells. We've also got ingredients in there to inhibit CD38, that inflammatory enzyme. So we've got apigenin in there in the form of parsley powder, which does that. I should probably point out we have a lot of actives that aren't put in their pure form, but in a more natural form. So things like parsley powder for apigenin, rutin instead of quercetin, and this is because they are much more bioavailable. It's fair enough putting pure active ingredients on cells and they work in cell culture, but when you try and take them orally, they don't do the same thing, and this is because they often get destroyed by the gut. So if you can put them in their more natural form, it protects them until they actually get to the cells. We also have inhibitors in there of a methylating enzyme, which we haven't spoke about, but quite often what happens in our cells as we get older is we get increased levels of methylation, which tries to also inhibit that recycling of NAD. So we have green tea in there, which contains EGCG, which also inhibits that enzyme. So, we like to talk of it as a multi-targeted approach. So every little thing that's going wrong in that NAD production network, we've got something in the product that's actually addressing it and trying to fix it. So a placebo-controlled, double-blinded crossover study is what we performed, which is the gold standard in terms of clinical testing. So double-blinded basically just means that the researchers performing the test and the people analysing the data didn't know which was the real thing and which was the placebo. The fact that it was placebo controlled, it's obvious what that does. It accounts for the placebo effect if someone knows they're taking something. The fact that it's a crossover study, what that means is that so we had 28 people in the study, and rather than them being split into two groups of 14 where half of the people only ever took the placebo and half of them only ever took the real thing, which is very often what usually happens, the fact that it's a crossover meant that every single person took the placebo. They then had a washout phase, and then every single person took the real thing. And some people, that they were split in that some people ended up having the real thing first, then a washout, then the placebo, and the other people had the placebo first and then the real thing. So what it means is that everybody is their own control. So rather than taking a group of diverse, biologically diverse people and on the placebo, then a completely separate biologically diverse set of people on the real thing, and then comparing them, which isn't a very good comparison, you can literally take a person and be like, this is what happened to them on the placebo, this is what happened to them when they took the real thing, and it's the same human being. So it's a much better trial design, but also a bit trickier and longer to do. But yeah, so we wanted to do this. Clinical trials are not required for supplements, believe it or not. The other thing about our clinical study is that we usually, for a clinical study in the NAD field, if you look at the trials on NR and NMN, they're all like, males age 55 to 65 years old, around that age bracket, which is not very reflective of the average population. So we actually used male and female, age 20 to 80. So again, that was a huge risk, because you would expect younger people not to have as low NAD and therefore not to see as big of a response. But we were like, this is something we're bringing out to a real, consumer mass market, that, we want lots of different people to benefit from it. And we want to test what's going to happen in the real world. So that was, another thing that's unusual about our trial, that we did that. But the things, we measured a number of different things. That the sort of primary outcome was, does this product actually boost NAD? And we found that after just 7 days of taking the product, it significantly increased NAD, and this continued to rise. We tested up to 28 days, but we're now doing some longer studies to see what happens on a longer term scale. So that was great that it did increase NAD. But the thing that we were really interested in was, are we actually fixing the root causes of NAD decline? And this is not just an effect of the precursors. So we actually measured levels of that key NAMPT enzyme, and demonstrated that after 28 days supplementation, the levels of this enzyme were significantly increased in all the volunteers, obviously compared to placebo, which was nothing. We also wanted to look at, inflammation. We know that, an issue with some of the other precursors is that they're possibly driving inflammation. So we measured levels of inflammation in all the participants, and we saw a significant reduction in levels of inflammatory cytokines across the study period. We also looked at, is NAD activating the sirtuins, because we know that a crucial part that NAD plays is in activating these longevity proteins. And again, we saw an increase in levels of the sirtuins. But I guess for me, probably what the most exciting thing was, the most exciting result was, we actually measured biological age. So there are tests now where you can actually look at the rate at which your cells are aging on the inside. And we measured biological age of all the participants, and before and after both the placebo and the supplement, and found that with the supplement, with Nuchido TIME Plus, we saw a reversal of biological age by 1.26 years, and that was in just 28 days. So for us, that was so exciting, because it showed that it really is having that impact on the cellular level to actually, at the end of the day, improve aging, which is what we're all about. But I think, in the near future, something that we are really focused on is looking at cellular senescence. We have things in the pipeline that we're looking to bring out to combat senescence. So senescent cells, very briefly, are cells that seem to become damaged with age. So our body puts them to sleep, and they sit, taking up space in our bodies and not performing their function, but also start to secrete a lot of inflammation. And there's been a lot of studies to show that if you can remove these troublesome cells, or if you can, stop them secreting the inflammation, then you can have a huge impact on health span.